Lopez Otin and colleagues proposed 9 hallmarks of aging, categorizing them as primary, antagonistic, and integrative. Cellular metabolism intricately connects these hallmarks, emphasizing the central role of energy regulation in the aging process. This review explores how mitochondrial dysfunction impacts these hallmarks and contributes to age-related diseases. Mitochondria are essential for life, playing a pivotal role in energy transformation processes within cells. Mitochondrial dysfunction can severely impair cellular energy conversion, particularly in tissues heavily reliant on mitochondrial energy production. Recent advances in aging research often operate within the realm of mitochondrial physiology, highlighting the close relationship between aging and energy balance. Calorie restriction, physical activity, and agents like metformin, resveratrol, and rapamycin can alleviate energy supply and demand imbalances. Calorie restriction's beneficial effects involve mitochondrial physiology, influencing OXPHOS, ROS production, and antioxidant defenses. The intricate regulation of mitochondrial physiology under calorie restriction interventions suggests that mitochondria play a crucial role in lifespan modulation. Suppression of the insulin IGF-1 signaling (IIS) pathway extends lifespan in multiple organisms, indicating an evolutionarily conserved mechanism. Studies in C. elegans have revealed extensive overlap in gene expression between IIS mutants and long-lived mitochondrial mutant strains. DAF2 mutants exhibit increased abundance of proteins involved in OXPHOS, increased respiratory capacity, and increased membrane potential. This increase in respiratory capacity is accompanied by increased mitochondrial ROS production, which induces expression of antioxidant enzymes, leading to an adaptive response. Inhibition of IIS improves mitochondrial bioenergetics and promotes a ROS-mediated adaptive response, contributing to lifespan extension. Mitochondrial morphology and dynamics are also affected by IIS, with DAF2 mutants showing mitigated age-related changes. In mammals, long-lived Ames dwarf mice and growth hormone receptor knockout (GHRKO) mice exhibit improved mitochondrial bioenergetics. Fat-specific insulin receptor knockout (FIRKO) mice also have increased lifespan and increased expression of genes involved in key metabolic processes. These findings highlight the complex role of the mammalian IIS pathway in regulating mitochondrial physiology and lifespan. Mammalian target of rapamycin, mTOR, is a serine-threonine protein kinase that regulates cytoplasmic, nutritional, and growth factor status, as well as cellular energy levels and overall cellular function. Inhibition of mTORC1 and consequent increase in autophagy has gained attention in the mitochondrial field. Decreased clearance of damaged organelles such as mitochondria is associated with aging and age-associated diseases. However, mTORC1 is also required for PGC1-alpha binding to the transcription factor yin-yang-1, which induces expression of nuclear-encoded mitochondrial genes. Modulation of mTOR activity can result in decreased mitochondrial bioenergetic capacity. In the context of mitochondrial diseases, inhibition of mTOR using rapamycin improves mitochondrial function and alleviates symptoms of Lee syndrome. Mitochondria may be the critical mediator for the effects of mTOR inhibition on longevity, representing a notable therapeutic target in aging and age-related diseases. Further research is necessary to fully understand the implications of mTOR inhibition in both health and disease. AMPK is considered the primary sensor of cellular AMP to ATP and ADP to ATP ratios, playing a key role in regulating cellular energy homeostasis and metabolism. AMPK activity is closely linked to mitochondrial physiology, with experiments showing that a reduced ATP to AMP ratio in skeletal muscle increases mitochondrial biogenesis via AMPK induction of PGC1-alpha expression. AMPK also regulates mitochondrial quality control including the autophagic turnover of dysfunctional mitochondria through initiation of mitophagy. Decreased AMPK activity may contribute to the reduced mitochondrial function and dynamics observed during aging. Sirtuins, a conserved family of deacetylase and deacylase proteins, require NAD+ for their function. In response to calorie restriction, the yeast NAD+ biosynthetic gene PNC1 is induced, increasing SIR2 activity and boosting mitochondrial respiration. In rodents, the age-dependent decline in NAD+ levels decreases SIRT1 activity, affecting mitochondrial homeostasis. SIRT1 regulates mitochondrial physiology by deacetylating and activating PGC1 alpha and FOXO, allowing control over a broad range of mitochondrial functions. Mitochondrial sirtuins also control mitochondrial physiology, with SIRT3 regulating ATP levels and AMPK activation. Failure to regulate energy homeostasis by these pathways is highly linked with mitochondrial dysfunction, leading to age-related metabolic diseases. The antagonistic effects of mitochondria have long been appreciated in organismal aging, leading researchers to view the organelle as more than just an energy supplier. The accumulation of mitochondrial oxidative damage, as suggested by Harman's free radical theory of aging, has been proposed as a driver of mitochondrial dysfunction. However, while some studies support this theory, others have shown that increased ROS production does not always shorten lifespan. Harman expanded the free radical theory of aging with the mitochondrial theory of aging, suggesting that mtDNA mutations may be the drivers behind the mitochondrial dysfunction observed during aging. The accumulation of somatic mtDNA mutations leads to a loss of mitochondrial function, resulting in a decline in energy capacity, increased oxidative damage, and ultimately cellular loss. Evidence for a contribution of mtDNA mutations to mammalian aging phenotypes comes from the mtDNA mutator mouse, which carries a mutated, proofreading deficient form of the nuclear-encoded mtDNA polymerase, POLG gamma. However, the role of mtDNA mutations as a primary driver of the aging process is still debated, in part because the number of mutations observed in normal age tissues is seemingly below the threshold required to affect mitochondrial function. Genomic instability in the nucleus can impact mitochondrial physiology during aging via anterograde signaling. DNA repair-deficient premature aging diseases have been linked to changes in mitochondrial physiology, indicative of physiological responses to anterograde stimuli. Nuclear DNA repair is energy-demanding, and persistent DNA damage can lead to mitochondrial dysfunction and metabolic deficits. NAD+ is a rate-limiting metabolite for the poly-ADP ribose polymerases, PARPs, which are key DNA damage response (DDR) proteins. An interplay between PARP1 and mitochondria may explain aspects of age-related functional decline. Deletion of the PARP1 gene in mice increases NAD+ levels, SIRT1 activity, and mitochondrial biogenesis. Cellular senescence is characterized by an irreversible cell cycle arrest, changes in gene expression, and a pro-inflammatory senescence-associated secretory phenotype. Senescent cells exhibit numerous changes to mitochondria, including their structure, dynamics, and function. Defects in mitochondrial OXPHOS are observed in the early stages of cellular senescence. Cellular senescence leads to decreased mitochondrial membrane potential, increased proton leakage, and increased ROS production, enhancing DNA damage and DDR. The contribution of mitochondria to the development of the senescent phenotype requires further elucidation. Epigenetic changes in metadNA have been linked to human diseases, including cardiovascular diseases, colorectal cancer, and neurodegenerative diseases. Methylation levels could influence where TFAM binds and affects the transcriptional response. Further research is necessary to elucidate the functional role of epigenetic alterations in metadNA in the context of aging and age-related diseases. Maintenance of healthy mitochondria and metabolic regulation is key to long-term health during aging. The metabolic syndrome, which occurs increasingly with age, is associated with obesity, hyperglycemia, dyslipidemia, insulin resistance, and cardiovascular diseases. Insulin resistance in older patients is associated with decreased mitochondrial ATP production and modifications in mitochondrial OXPHOS. mtDNA polymorphisms have been associated with an increased risk of metabolic diseases. T2DM is a chronic disease characterized by dysregulated blood levels of glucose, impairing insulin action and insulin secretion. Mitochondrial dysfunction and the associated impairment in fuel oxidation are notable contributors to the development of insulin resistance, non-alcoholic fatty liver disease, NAFLD, and T2DM. Decreased mitochondrial oxidative capacity and mitochondrial content are seen in skeletal muscle from patients with obesity and insulin resistance. Beta cells respond to increased metabolic demands by increasing their mass and insulin synthesizing and secretory activity. Long-term and chronic exposure to hyperglycemia can cause desensitization and even depletion of beta cells. Mitochondrial dysfunction is increasingly thought to be a central contributor to beta cell failure in the evolution of T2DM. NAFLD is the most common form of chronic liver disease, with epidemiological studies suggesting that it may be a precursor to the development of other conditions, such as insulin resistance and T2DM. Patients with NAFLD present with decreased ETC complex activity and a fatty liver-associated decrease in SIRT3 activity. Vascular aging is the major risk factor for vascular diseases and health status in the aged population. Oxidative stress, a characteristic of T2DM, hypertension, and atherosclerosis, is also the dominant feature of vascular aging. Elevated ROS levels lead to endothelial dysfunction, an early event in atherosclerosis pathogenesis. Mitochondrial dysfunction is a hallmark of many chronic inflammatory diseases. Several alarmins consist of mitochondrial molecules, and the ability of these molecules to activate PRRs is dependent on their similarity to bacterial components. An uncontrolled release of mitochondrial components can induce chronic inflammation, a condition associated with several diseases such as rheumatoid arthritis, atherosclerosis, liver injury, obesity, and aging. Exercise is a well-known lifestyle intervention that prevents metabolic disorders, such as obesity and T2DM. Calorie restriction is another widely known lifestyle intervention that can prevent age-related metabolic disorders in multiple species. Several polyphenols that improve mitochondrial physiology have been described, including resveratrol, which enhances mitochondrial biogenesis and oxidative capacity. Metformin, widely used to treat T2DM, activates AMPK, improves mitochondrial function, and decreases age-associated inflammation. Systematic NAD+ decline and decreased sirtuin activity are considered among the molecular events that occur during aging. Supplementation of the precursor nicotinamide mononucleotide, NMN, re-established beta cell glucose-stimulated insulin secretion, as well as hepatic and skeletal muscle insulin sensitivity in mouse models with impaired glucose homeostasis. Mitochondrial uncoupling is defined as a process that uncouples nutrient oxidation from ATP production. The mitochondrial uncoupler 2,4-dinitrophenol (DNP) has shown potential therapeutic benefits in obesity and metabolic diseases. Mitochondrially targeted coenzyme Q, MitoQ, or mitochondrially targeted vitamin E, MitoVit-E, have been shown to target mitochondrial dysfunction. MitoQ protects against oxidative stress and offers neuroprotection in preclinical studies. The rate of aging is coordinated, at least in part, by conserved genetic and biochemical pathways, many of which control mitochondrial physiology. The modulation of these pathways could be the key to improving human health and preventing major age-related diseases, especially those associated with metabolic dysfunction. Despite continuing progress, combating mitochondrial dysfunction in age-associated metabolic diseases has proven challenging. New digital health technologies, such as artificial intelligence and big data, could pave the way to understanding the complex nature of these diseases, providing associations between specific biological or clinical phenotypes and corresponding biomarkers.